Welcome. Carly, take it away.
Hi, everyone. I'm Carly Kislisnik, the project manager for OpenScope over at the Allen Institute. And thank you guys all for joining today. We're going to get started in a second with a brief intro to the agenda. And just a heads up, you guys can please introduce yourself in the chat at any time. But if you have any questions along the way, please use the Q&A box down at the bottom, and we'll either answer them in writing or answer them live during the Q&A section. So just a reminder, chat to introduce yourself and Q&A box for any actual questions. All right, I'm going to be handing it over to Jerome to give us an overview of the agenda for today. Jerome, if you want to go ahead and start sharing your slides.
Let me do that. All right, so good morning, good afternoon, everyone, wherever you are. My name is Jerome. I'm an associate investigator here, heavily involved with the OpenScope project. So today we're going to have our annual webinar where we introduce the OpenScope call for experimental proposal. This is where we can detail the call and also answer questions. We also have a guest today, Gonzalo Lopez. So the goal of the OpenScope project is to build a community around the observatory, as much as sort of serve experiment to the community. For me and for the rest of the group, it's really important that we support this community, the ecosystem around observatory, so we gotta help nurture it and develop over the years. So there are many components to this community. There are teams that develop data analysis tools, there are teams that develop new technologies. And Gonzalo has been leading effort to build a behavior software platform that has been growing in the community in the last decade. He started this work at Champalimaud in Portugal during his PhD as part of his research on behavior. I encourage people to look at this PhD manuscript. There are some interesting observation on the motor cortex that I've enjoyed reading. But on the development of the software, that became actually very important because the community has been growing tools to peer into behavior, and it's very important that we control behavior way in a standardized and an easy to reproduce way. So Gonzalo pushed that. He actually developed a new programming language and then moved to do his postdoc and keep pushing that. Now he's heading a company called Neurogear. And we are interested in adopting Bonsai in the future. So we have been working on testing and integrating Bonsai into a pipeline. This is ongoing. This year, since we are opening up the behavior capability in OpenScope, I felt it was important to highlight the development on the behavior front end and in the community, and to use our platform to give them attentions and so that people could see what's happening in the behavior ecosystem. So that being said, I'm going to give the floor to Gonzalo, and then we'll talk about in the rest of the agenda about the OpenScope RFP at 9:45 approximately, once Gonzalo has given his talk. And then there'll be Q&A on the way, as you can see on the agenda on the slide. So Gonzalo, I'll let you take it.
Thank you so much, Jerome, and thanks for inviting me. It's great to see initiatives like the OpenScope, starting and pushing thinking about behavior and neuroscience forward. It's really important. Let me try and share my screen. See if can you see slides?
Looks good.
Okay, great. So I'm going to talk a bit about a vision that's been developing. There's a lot of introductions to Bonsai that I give, but this time I tried to kind of take it in a slightly different direction from usual, because I think the next couple of years in neuroscience, and especially in open source neuroscience are going to be really exciting, and I wanted to try and share a bit of that vision with you. But yeah, we can happy to take it during questions in any direction that you are interested in. And yeah, Jerome, I think will share a few links about resources where you can learn more about the language. Yeah, so but just as a way of introduction, quick introduction, yeah, as Jerome said, yeah, my original background is in computer science and engineering. I moved to neuroscience, but what I was doing before moving into neuroscience was actually designing computer languages and frameworks for augmented reality, virtual reality, and real-time sensor processing. And when I basically moved to neuroscience, one of the things that I became immediately aware of was that a lot of the research and work that I was doing before was actually immediately applicable to studies of behavior and integrating hardware, because that's what you need to kind of relate signals from the brain to what animals are actually doing in their natural environments. And yeah, I became really fascinated by that application, and that was kind of like the seed that kind of got Bonsai growing, is basically taking that experience of trying to get basically technology in an easy way in the hands of people who want to do interesting stuff with computers. That's basically the main motivation, but now applied to neuroscience. And yeah, so what I will try to talk about today is how that's going. And to that, I'm going to give a very brief introduction on the evolution of behavior paradigms in neuroscience and why they're important, and what kind of revolutions they brought about. And then I'm going to describe the current state in the toolkit for pushing behavior paradigms towards a more natural behavior. And then I'm going to then try to outline, yeah, the current vision we have for where we think we are at the cusp of transitioning into kind of prototyping and designing new experiments going forward. Yeah, and yeah, if any questions you might have, I won't be monitoring much the Q&A, but yeah, if you want to stop me at any moment with questions, I'm more than happy to stop and address them. All right, so the first thing I want to talk about, yeah, is this kind of evolution of behavior paradigms. And one thing that I always like to think about is sometimes the biggest revolutions actually come from a change in perspective, from looking at concepts we already think are familiar. And there's lots of ways to do that with behavior. One of them that I think is very fruitful is this one by William Powers. In 1973, he introduced a new way of thinking about behavior, not so much as an input-output mechanism, but rather as a control process, where behavior is basically the control of perception. And what this means specifically is perception here is interpreted as the entire sensory state of the animal as interpreted by its brain. So not just the sensory stimulation, but actually the entire, like how the brain builds the internal representation of what that state is, and including the desires, the values, the motivations, memories, and so on. And then the idea of this being a control process is what the goal of behavior is basically to bring that perception. So what state am I in as my whole behavioral state? Is it in a desired state or not? Like, do I want to be thinking the kinds of thoughts that I want to be thinking, and the things that I expect to be able to do and want to do? Is that's kind of like the goal of behavior. And the reason why I think this is very interesting is because I think actually a lot of the revolutions that have happened in neuroscience over the years have come from basically interpreting the relationship between animals and their environments actually in the light of this, which I find particularly illuminating. So this introduces essentially a relationship as it's drawn in this box, like of the agent and its environment as a loop, a closed loop, where the agent is the animal is trying to interpret the sensory state, and then is trying to kind of do actions on the environment such that the state changes to something that is desirable. And yeah, so to kind of put this into context, why I think this has been like very fruitful for systems neuroscience and why it's been revolutionizing as we move basically towards really moving behaviors and natural behaviors. It's kind of like a a kind of an axis of control that we've been traversing, from basically having animals that started out anesthetized, and we would present the stimuli to them, towards then them actually being awake and experiencing the stimuli, but still head fixed, and then finally moving to kind of a freely moving behavior paradigms. One of the things that people report a lot from moving to freely moving behavior is like a loss of control. And this is quite interesting because I find it interesting, especially under this paradigm, because basically I think there's like two axes of control we're talking about here. So one is the control of the animal over its own perceptions, and the other is the control of the experimenter over those perceptions. And yeah, the idea here is actually there's these two play a tug of war with each other. So the experimenter wants more control over the perceptions of the animals, but the animals, they want to do their own thing. And there are ways, what I like about this kind of way of thinking about behavior as the control of perception is they provide ways of kind of bridging the gap between that tug of war. So one of the ways, just to give a few examples, then we'll move on to where we are currently in terms of tools that allow us to do this. One very popular one, well, one of the most popular stories in this space is, of course, hippocampal place cells. And one of the things about, this is just a video of an animal navigating a linear track, and probably many of you are familiar with this, where I think you're hearing also the sound of this. Okay, take it off for now. Yeah, you know about the story where basically animals have this representation of space in terms of coding for individual neurons will code for certain locations in space. And what's nice about this story is that it started with basically a loss of control of let's just let animals roam freely in an environment and record from their brain activity and see if we can make sense out of the recordings that we get. And usually the story goes that there's this kind of inspirational moment where we leave animals kind of roam freely in the environment. But actually, if you step back from that story a bit, one of the main success elements of that story is actually to interpret the data correctly. You needed to bring back the element of freedom of the animal back to a controllable state. And the idea, what you do in place cells is literally you actually fixate the animal, the data about the animal's brain state by fixating, you have to record everything about the animal's movements in the box. And then you ask a question, which is, what does the activity of this neuron look like when the animal, at the moments where the animal passes for a specific location in space? And basically that you're kind of in a way fixating the animal virtually in that space. It's just that the animal spontaneously happens to traverse the states that you're going to interrogate later. So that's a very interesting kind of observation where basically you can recover from the loss of control as an experimenter of letting the animal run freely, basically by recording in detail the degrees of freedom that you've lost, and then kind of post hoc recovering those degrees of freedom by in analysis, where you ask, now I want to fix the location in space, and I want to average my data around that fixed location. And even though the animal was roaming freely, that is how you recover these kind of place fields. So that's one way of kind of getting control of your perceptions, even though the animals can feel free to kind of choose them as they feel like. The other approach that has been very successful with freely moving behavior is basically what I call sometimes voluntary fixation, which is a kind of operant conditioning. So the animal kind of, we create situations for the animals where we make it attractive to them to choose certain perceptual states over others. So we kind of we introduce these reward conditions. We present stimuli to the animal, and it turns out that they're motivated to get satiated and to get rewarded, and then they themselves choose to put themselves into predictable conditions. So that's another way that you can kind of take control over, even though they're very much moving freely, but you let them make the choice that puts them in an interesting state, easy state for the experimenter to kind of measure. And the final the final kind of way of tackling this problem of freedom in the state is, I call it informally like interactive fixation, is like a closed loop setting where basically you're taking control over both perception and action. So you have like an environment that is interactive. So it's the same way that the animal is engaging in an interaction with this environment. Now we have a dynamic environment that is also engaging in an interaction with the animal, and we can measure both sides of the equation. So both the perceptions or the stimuli that are arriving at the animals, and also the kind of in close tandem the actions that is taking, and then you can modulate basically the link with how the actions of the animal generate the perceptions. So that's where we want to kind of the strategy that I think is very nice of kind of like allowing animals to kind of grow more and more naturally and free, but still keep track of kind of like the control, how to make your data like analyzable kind of at the end. So now, why this I think is, one of the things that was motivated me during my own research on how we try to kind of think about ways of promoting more natural behavior studies in the lab. And this is all doable, and all these studies have been kind of pursued in very fascinating directions. Still, it's not done enough. And what I think, what we think is kind of there's several obstacles to this, and definitely one of the biggest obstacles is it's not easy to set up. So it's like it takes a lot of time to develop a behavior rig that kind of records the behavior of the animal in enough detail, and it kind of and that kind of thinking through all the steps that are necessary to kind of set up the questions in this way is a huge investment, and it turns out that it's also usually very hard to make it general so far. So it's it has to be often tweaked to specific situations and specific questions. And so all the investment you've put in into one of these behavior rigs, of course, in the case of place cells, you can actually explore a lot of variations, which is nice. But in many other situations, it's actually much more difficult to kind of start changing the assay once you have it running. And that's has also pushed people from kind of putting the investment in to kind of keep these kind of natural behavior rigs in the lab, and because they require also a lot of different hardware and software to get things right. They're also hard to maintain and extend because it's there's a lot of this is connected to the second point about the assay being specific to a situation, scientific question. And then, all these 3 points are kind of related. It's also hard to share and reproduce for the same reasons. Now, what I'm going to argue about today is that this is one of the main reasons why open source neuroscience has become so popular in the last decade or so. Is because it was built basically is the perfect ecosystem for promoting this kind of approaches to behavior. And the reason is because one of the main pushes for open source neuroscience is not just that it's kind of open source and free, but actually it's the idea that a system is built to be modified. And that's one of the most important aspects of open source neuroscience, is that all of these projects that I'm listing here were made with the ethos that basically you're going to take a technology that is very useful for integrating to your assay, but actually you want to integrate it in such a way that it can be modified, it can be interoperated with other hardware and software, and it's made to be tailor suited to your specific needs. And it has worked out so well that now, basically with a collection of just a subset of these tools that we have here, sorry, just go back. We're now exploring basically all of the assays that are most popular in kind of freely moving neuroscience can be explored with this kind of open source hardware and software. So what my role has been in this, as I was describing, is Bonsai. So one important aspect of making sure that the systems are interoperable and kind of can be tweaked and modified easily, as I was describing, is to have a way of combining the different tools, because each of them is kind of specialized for a specific purpose. And the goal of Bonsai is to kind of give us a language for experimenting with those tools and creating systems that are integrated. Because again, when you're dealing with these kinds of systems, you need to have an integrated kind of system. Is much harder to kind of keep things separated. You can, but it at some point you'll start really you start losing a lot of the possibilities. You start to have like blind spots in the map of possible experiments you might want to design. So Bonsai is made to kind of help fill that gap quickly. So it's basically a compiler, a standard library, and a development environment where you can visually compose data streams in real time, and each of these data streams can be independently controlled, independently streamed. They're not tied into any specific kind of ecosystem. So you can combine like an OpenEFIS with a Miniscope, with closed loop real-time computer vision, with machine learning, with Deep Lab Cut, with real-time hardware for controlling behavior hardware. And it's been quite interesting to see for me how much this has taken up, as Jerome was saying, over the 10 years. So Bonsai is this year is doing its 10th anniversary. And we've basically seen people taking Bonsai to do all kinds of freely moving tasks, which for me is a mark of success, because it's exactly what it was designed to do. And the nice thing is you can do it without changing a lot of the hardware that is involved in these tasks. So I've seen like labs that started with like a head fixed behavior task, and then they start asking questions in the forums about, oh, can I just take the same cameras and the same machine learning software that I use to track the body movements of the animal and use it for an open field task? And yes, you can do that. You can just literally, in many cases, you can replace the logic portion of the task and keep the same hardware, but now make a box, put the animal inside, and still run the same optogenetics driver, still use the same camera you're using for eye tracking, but now you use it for tracking the animal in the box, and it's quite easy to do that. Okay, so I'm very happy with where we've got. But what I wanted, as I said, what I want to talk about today is basically where we're going in terms of expanding now this neuroethology toolkit. And I'm basically going to talk very briefly about three main directions where we're pushing this space. So one is, we want to change. There's a lot of like tedious chores that are kind of in our way when we're analyzing neural data, and all these points are kind of made to address them. So one is in neuroscience, in behavioral neuroscience and systems neuroscience, time is usually everything, because it's one of the main, because we're looking at temporal correlations, is one of the main measures of one of the main sources of data that we care about. And we spend a lot of time basically aligning data streams, like synchronizing stimulus to brain signals, synchronizing the camera position of the animal in the camera to those data sets, and so on. And we also want to argue that, similar to what happened with place cells, we want to do the same. We want to get rid of basically all the logic all the time we spend aligning synchronizing that data. And we also want to get rid of, do the same thing for space, actually. That's the other thing we want to move more and more towards, is brains care a lot about not just time, but also space. And we believe it's been neglected a bit. So we want to provide tools basically to make it trivial for us to analyze also data across that dimension. Then the other thing we want to do is make it even easier to combine systems at scale while guaranteeing reproducibility of data, to make sure that it's really easy to swap out hardware modules, task logic modules. You can design your own tasks. upload them somewhere, and then you'll be able to kind of take advantage, leverage like an existing hardware infrastructure and completely reconfigure it like very easily and still have guarantees on the data in terms of, again, synchronization and the quality. And then we want to finally have Bonsai promote a culture of moving a lot of the analysis pipelines that we run, including spike sorting, tracking, inference of state, to actually run in real time. And that's something that, again, driven by this push for controlling in real time the state of the animal in this kind of closed loop way. We need this for really pushing behavior forward. Okay, so just briefly to talk about how we're doing that at the moment. So we have been in the last few years developing this hardware ecosystem together with the Champalimaud Foundation and the OpenIFA's production site, an ecosystem for timestamping every measurement and every stimulus that is controlled by Bonsai in hardware. And this is basically to get rid of all, as I was saying, all of that synchronization logic that we have to do. So basically what this is a board or family of boards that are integrate neatly, directly, seamlessly into Bonsai. And they're taking care of all the timing, all the real-time control commands, and also every measurements we're making, they're being time-stamped in hardware. Again, not just the measurements, but also the commands. So if I ask an LED to turn on, the board will timestamp it in hardware when the LED actually turns on, not when the computer asks for it to turn on. And the other really nice thing about these boards is that they can self-synchronize their clocks automatically. So you can have like multiple of these boards. They will share a clock signal across them, and they will adjust their timestamps such that when you collect data from all these different data streams, the timings on those data are already immediately comparable. There's no need to kind of go back and count TTLs and look at measurements from compare, like find some logic to compare the events between two different devices, because that will be solved at the level of the hardware by these boards. So in that way, we basically get a completely time-stamped, like a unified view of time, basically for Bonsai, which is really nice because Bonsai is not real time, but these boards are. So it's kind of takes care of that for us. So that takes care of time. Then for space, we are working basically, we started integrating, of course, one of the main challenges when you have freely moving animals is you need to keep track of where different parts of the animal are at any given time. And of course, tools like the lab cut have been exceptional advances in this kind of problem space. And they've given us a lot of kind of control over this problem in a way that is easy. I mean, you could do this already with various other ways, but it just opens up democratizes that the solutions to that problem in a much nicer way. And also, we have solutions coming into Bonsai, for example, from systems like Onyx, the next generation OpenIFA's acquisition board, where you have head stages that are come equipped with 3D tracking systems on the head that can give you like a very low latency reconstruction of where the animal is in a kind of a complex space. And these basically allow us to have the beginnings of a common spatial reference for our data. And not just for the data of the animal, but also for the stimulus. So, because when animals are navigating these dynamic environments, we want to be aware of basically where the stimulus are being presented relative to the animal's receptive fields. Like if I'm showing if I have a screen on the box and the animal's moving freely relative to that screen, I want to know where exactly in the animal's retina the stimuli are impinging. And to do that, I need to keep track of where the animal is. I need to be rendering the stimulus in a way that is consistent with the position of the animal. And if I do that, then I will be able to get that kind of control in a similar way as I was discussing earlier with the virtual fixation in hippocampal place cell research, you can do the same for freely moving vision. That's the kind of hope that we're kind of trying to build towards in the next few years. And then we want to get all the way also to neural recordings. So also in terms of spatial reference. So we want to know exactly where we put our probes. So we've been designing systems where you can kind of, during implantation, keep track of where you think the probes you've implanted are at. And also in real time, you can reconstruct, produce estimates of basically where in these dense probes that are very nice because you can kind of grab from many channels at the same time. You can kind of reconstruct very nicely where units are, even without the traditional spike sorting often. And that the hope with that is we can also localize exactly where the units are in real time as the animal is running through the environment. Again, in the spirit of bringing everything in a kind of real-time pipeline. Okay, and so that's it for space. And then the final step, as I was saying before, so we want to make sure that we keep the spirit, the possibility of rapid prototyping tasks and sharing, making sure that those tasks can be shared. So we've been working gradually towards standards for specifying behavior tasks in Bonsai. And the main hope here, I mean, there's lots to say here that I won't have time to go into, but our main goal is, as I was hinting at before, is that you can kind of have a more or less intuitive translation of a diagram that you might have of a task. Let's say in this case, a go, no go task, where you have like after each ITI, you have like some ratio of like go and no go trials, and you have like specific outcomes that can kind of flow from that task structure. And we now have ways of basically in a relatively simple, compact form to translate those into Bonsai, a kind of a graphical manner. But then the nice thing is you can also make them independent of your hardware that you're running, such that you can simulate the task logic even without the rig. You can kind of compose a task structure that you care about, and then you can plug it into hardware that is specific for certain rigs, which means it will be much easier to kind of share certain features of abstract task structure across different labs. And also for logging and monitoring, we have the same philosophy. So we're trying to basically design visualization modules in Bonsai that can be kind of plug and play into different statistics you're measuring from the tasks. And it'll just speed up the way of interpreting and debugging those more complicated task structures as you're running them. And finally, this is all also powered by a maybe sometimes less known feature of Bonsai, which is you can actually deploy self-contained environments in Bonsai with all the dependencies that are needed to run the task in simple text files that can be stored together with your task, so that you can make sure that you can kind of share a GitHub repository, an open source repository with all your task description. And very similar to how Python stores its environment descriptions, you'll be able to bootstrap an environment that is ready to run the task logic from these files, and make sure that whoever is receiving your task has everything they need to kind of get started. Then we're also building finally interoperability with more and more external systems. So we're expanding the range of protocols that we can accept in Bonsai, including 0MQ, lab streaming layers, open sound control that we've used before, and also the range of languages that people have been interoperating with Bonsai. So more and more we've seen people basically take MATLAB Python tasks and basically use Bonsai as like kind of a server for presenting stimulus and controlling certain dynamic aspects of tasks, and such you can kind of combine, kind of do hybrid systems that are at least for people kind of transitioning to the realm of freely moving behavior, have turned out to be a really nice kind of stepping stone for them to start exploring that space. Yeah, and just to kind of finish up, because I also want to kind of give time for questions, we've been putting a lot of effort recently on growing documentation and learning resources. Yeah, and I guess Jerome, I also share these with you later on. Here are some links of, we've been putting a lot of effort in finally getting Bonsai docs up to speed. So the new documentation website is online now with tutorials and the reference manual for all the main core operators. And we're also organizing in collaboration with the Cahal Advanced Neuroscience Training Program courses on Bonsai that actually you can apply to the latest one right now. But what we'll be doing is actually bringing content from those courses back into the documentation website. So hopefully our hope is that a lot of the learning gap that existed in Bonsai will be kind of hopefully made much smoother, the learning curve over the next couple months. So that's where we're going. And yeah, here's like a list of like kind of coming soon features, but we can discuss them in the questions and answers. I think it's I don't want to take like too much more time, but yeah, there's a lot of stuff to be said about how all the things that I was kind of hinting at are taking shape and becoming very, very real very quickly. And yeah, hopefully we can have like a nice chat about whether this is all, how far are we from actually having this kind of absolute control over space and time and being able to move towards these ambitious experiments. Yeah, thanks everyone that has helped make this vision possible. So we now have a bigger and growing team behind Bonsai, and we're collaborating a lot with a lot of other open source projects. We really want to build like a shared momentum behind these technologies, as also the Allen are doing and supporting. And it's really nice to kind of hopefully seeing this community come together and really leave a lasting impact on the field. So yeah, I'll leave it at that and take it from here.
Thank you, Gonzalo. That's very informative. And we do have a couple of questions coming in. And just FYI to everyone, we are moderating the questions in the Q&A chat on Zoom and then also the live stream on YouTube. So a couple questions. One, someone was asking that, is there a plugin in Bonsai which will communicate with NeuroPixels probes? Someone has been using the ITAN compatible RHD 2000 node to read INTAN data, but unsure about NeuroPixels.
Yeah, I can quickly answer that. So the answer is yes and no. So the yes part, if you can still see my screen. So there's a package that is available in the package manager. I'm not sure if it's in the community, NeuroPix. Yeah, so there's a Bonsai NeuroPixels library that you can install. The no part is that this is only for the preview version of NeuroPixels. So before NeuroPixels 1.0 even, there was a dev board, I think NeuroPixels release version 3A, preview 3A or something like that. And this was made, as you can see, it's from 2017, the package. It's actually quite old. The good news is we're working with, well, there's actually another route you can take is essentially use the Onyx system. That's what I was going to propose. So depends on what system you're using to get data, but the next generation OpenEFIS data acquisition system supports NeuroPixels 1.0 head stages, and that is fully compatible with Bonsai. So you can just plug and play into it and get your data. But we'll also keep making efforts to supporting the just bare bones PXI-E chassis over the next few months. I think we'll try. But yeah, these are currently the available options you'd have for you, depending on which system you have. But yeah, if you have a PXI, yeah, the bad news is if you have a PXI chassis, then nothing out of the box right now. But if you have an Onyx system or an old PCI Express interconnect, the preview boards, then yes. Yeah. But it's possible, yeah.
Great, thank you. follow-up to that, we have someone asking, is there a way to have OpenEFIS GUI synced and operating through Bonsai? So ideally, so that once I start my Bonsai, OpenEFIS GUI is opened, and when I run my program in Bonsai, the OpenEFIS starts recording too. I think it would be great to be the signal live during recordings.
Yeah, this is great, actually. So actually this, there's something I haven't explored yet, but I just realized the OpenEFIS GUI actually already now has a new feature, which is an HTTP server that allows you to remotely control a whole GUI from external applications. So this is something we haven't explored right now, but in theory, you could right now design a plugin for Bonsai to start an OpenEFIS recording session remotely. If I understand correctly, how the capabilities of that plugin, of that new feature in OpenEFIS, the GUI 0.6.0, I think, will come with that HTTP server. So that will be something that is very worth exploring. And also they've been adding new functionality for streaming raw data out of the OpenEFIS plugin into something like Bonsai, either through lab streaming layer, for open sound control. There's new plugins. If you go to the OpenEFIS, actually the same, but with the, I think it's just the GUI docs. I think these have been revamped recently. And if you look for, I think, tracking plugin, but I think there's also the, if you look through here, you'll see ways of getting the data out. I think they're working on that like literally right now.
Great, thank you. We got a lot of questions coming in. Another one is, what is the typical time delay for closed loop experiments? And is that delay fixed, or can it vary or vary unpredictably? So what I got from the talk is that it seems that the hardware from HARP can run in real time, but for example, spike sorting and triggering optogenetic stimulus in response to a burst from one particular cell would run via Bonsai, and thus not in real time, question mark? What would be the time delay in that case? Yeah, exactly.
The main question is exactly that, the time delay. So real time, it depends on what latency you are after. So one thing we get with the HARP, just native HARP, you get 1 millisecond fixed latency, like either way. So you get basically a 2 millisecond round trip time, which is pretty nice for behavior experiments. But if you want to do some kind of faster, closed loop stimulation, it might not be so ideal. With the Onyx system, you get really exciting timing results, actually. So one of the main trade-offs that the Onyx system had in relation to the PXIe like the PXIe version of NeuroPixels, for example, is they've put the scale more towards the low latency side than the buffering side. So you can do now closed loop through Bonsai below a millisecond, actually, with data that is streaming live from the Onyx system. It basically runs, the trick here is to basically let go of the USB, because most of these latencies that we experience, they come from communication latencies or from limitations of the refresh rates of the devices that we are plugging in. So for example, what they did with the Onyx is they let go of the USB interconnect at all and just use the PCI Express card that can just reduce the latency tremendously, almost by an order of magnitude compared to, let's say, the PXI system. So if you do that, then you can do, they've been able to basically run experiments where you can, before the spike is even completed, you can close the loop and stimulate the cell going through Bonsai. So that's kind of quite impressive results that they're presenting, which is really nice. And for example, also in other modalities, just to finish this question, for screens, so closed loop visual stimulation was often limited by the refresh rates of screens, which is usually like 60 hertz. But now with these like ultra high refresh rate with gaming monitors, you get screens that can do 244 hertz. And now we're getting like latencies with Bond Vision, for example, we're getting like 8 milliseconds. If you take like a G-Sync card and these high refresh rate monitors, we're starting to approach the 10 millisecond kind of mark for visual stimulus kind of response, which is again really nice compared to what you could get just a few years ago. So I think the space for like reducing the latency, even going through a system like that is non-real time like Bonsai, just because the sheer communication speed is so fast, it's getting much more exciting space even for that. Yeah.
And I believe this is a follow-up to that question. So is that including clustering or spike triggered on threshold, and millisecond range is great?
Yeah, this is not including clustering. Yeah, this is just spike triggering on threshold. Yeah.
Perfect. So we're running out of time. There's some additional questions here, which Gonzalo would be great if we can help type some answers to some of those. I'm going to follow up with two final questions. One, is the training course free, and what are some additional resources people can look for this?
Yeah, the course is not free, but you do get a nice perk out of it, which is you get a whole kit. So the course registration comes with an actual hardware kit of hardware you need to run the course, like includes cameras and Arduinos and a bag of sensors and actuators. And on registration, you get that for you to do what you like. So that's hopefully, and we try to keep the whole mission of the Kahal is to make these online lectures accessible. So hopefully it will not be much. I think I don't know if they should have the pricing on the website. Yes, for group. Yeah, they have two. You should go through the website, but they have like a registration fees for person and for groups. We're running lectures now, courses that are live in different locations worldwide. You can look at the website for more details, but it's kind of an interesting new model.
Great, thank you. So we do have a few other questions in there. We'll try and type responses, but there is additional time at the end for general Q&A after the OpenScope section as well, if anyone has more questions there. And one last question that might be answered best by both you and Jerome and lead into our next section is, how do Bonsai and OpenScope relate? So just a reminder there. So that'd be a great quick question to answer as now we lead into the OpenScope section of the talk.
Maybe Jerome, do you want to answer that? I don't know. Do you?
Yeah, so we've been collaborating with Gonzalo and adapting our current detection of change task to Bonsai. It's an ongoing process. And so our vision is that in the coming years, we'll be doing more and more Bonsai behavior, because it opens more open source capabilities and integrate with the ecosystem. So that's why we have him here.
Great. Well, with that, we thank you very much, Gonzalo, for giving us that overview. Lots of great questions. And again, we'll be around later for additional Q&A and try and type out responses. And now we're going to move on to the OpenScope section. So if Jerome, you want to go ahead and load up our slides for that section. And now we're going to be covering the OpenScope opportunity, kind of the mission and goals of OpenScope program at the Allen Institute, as well as specifics for this year's 2022 RFP and some things that may have changed from last year. So with that, I'm going to let Jerome take it away and to the next part of the agenda.
All right. So let's speak to the OpenScope RFP this year. So this talk, there's a couple sections to it. As in previous year, we're gonna talk about the overall vision and also the pipeline that is behind it. This is we've talked about it this year, but we thought it was still important to speak to it because there might be new comers to the program. And then we'll have a section about the specifics of the 2022 request for proposal. Now, there are some changes from last year that I want to speak to. And based on this development, we'll finish with future outlook and development that we want to push to the program in the remaining years of the program. So why do we need an observatory in system neuroscience is an important question to answer. And our vision for this was really inspired by the astronomy field. This is a vision that Christoph Koch brought to the Institute with him, and we built the pipeline with that intent from the beginning. If you look at this publication from the Keck Observatory a few years ago, where they did some analysis of the scientific impact of this observatory that is shared with the community. On the y-axis here, there's the number of publications that are coming from this platform. You see that as the platform is made available, the community is building up its use, and to the point that a decade later, as many as 300 publications came from out of it. So the impact of such a resource on the community can be really important. And we felt that the current academic model in neuroscience to have like each individual laboratory build their instrument and publish after that, and then sometimes the instrument is being immediately deprecated at the end of the PhD, might be sort of a detrimental, sort of like iterative improvements and building a platform that push the boundaries of technical development. And so we felt that there was a potential need for an observatory in neuroscience because the technical development is getting increasingly complicated. And that limits our ability to push the boundaries of what we can record from the brain, which is sort of outlined in these slides. And that's important because we know that a large number of experiments are needed to understand how the brain works. Behavior is diverse, but you need many, many experiments to understand how it works. But if these experiments are never integrated with one another, we can't sort of build a whole picture of how it's performing. And so an observatory has the potential to sort of centralize some experiments that are essential to the community and make this analysis, this meta-analysis possible. In addition, some experiments are actually entirely duplicated across the community. So it will be better to just perform this experiment and then make the data available to the entire community and then have the lab sort of use this as seeding insight to sort of explore the diversity of the brain computation. And lastly, and importantly, we think that an observatory model can actually synergize the main community in neuroscience. There are experimental neuroscientists that perform experiments with animal models. There are technical neuroscientists. Gonzalo is one of them. We built the tools to make this possible. And theoretical neuroscientists that pose questions, computational questions about that should be performed in the brain and need to be tested by the rest of the community. So these three groups are needed to answer questions on the brain. But each one of these categories is requiring a lot and increasingly more sophistication and time. And as we push the boundaries of technologies, you can't be the best on all fronts. And we think that an observatory allowed to sort of make a place for technical development to be disseminated. So allow the technical neuroscientists to develop their tools and have a way to distribute it. Make it available to experimental neuroscientists to sort of build projects and use it to seed over project, and also give access to a theoretical neuroscientist. So we think it's an opportunity to sort of create a community that interact well with all the sub-community of the neuroscience field. So that's when we started to build the OpenScope program. And in the last few years, we've sort of pioneered this with a couple of pilot attempt at building this observatory. And a year and a half ago, the NIH kindly agreed to fund this resource as a U24 for the community. So it's a pre-funded resource. So it's essentially given access for free to experiment. This resource is part of the MindScope program at the Allen Institute. And our goal is to open up 2-photon and NeuroPixel experimental platform for experiments that are proposed by the community. In addition, we also have, as I said, long-term goals. One of them is to maximize the scientific impact of our platforms, to reach beyond what we initially intended in terms of scientific questions, but also to build an inclusive community. That's why we have Gonzalo today. And also develop this ecosystem around the observatory, which is illustrated on this picture on the right. We also have a couple of guiding principles that sort of guide us towards all the decisions we make. We are built for the community. That's really important. Another aspect is we want to empower the ecosystem, not replace or rebuild it. So whenever possible, we'll synergize and leverage existing tools. And lastly, open science, open source, standardized protocol. This is really our core DNA, our core values. And so we aim to encourage and disseminate these practices. And I'll speak to that in some slides in the talk. So there are a few components of this resource. The first is an access to the Allen Brain Observatory data collection pipeline. So this is a hand-to-hand pipeline from animal husbandry to brain reconstruction, data collections, and packaging. That's the first component. The second component is this yearly proposal cycle that we're advertising today with double-blinded review. And it's open to the entire community. The third component is integrated cloud deployment and analysis ecosystem. So we are partnering with the NWB and the Dandy team on this, and we're hoping in the years to come that this is going to grow and be more useful to all of the projects. And lastly, but importantly, a technical hub. And so that's a place where people can come and create community, create connections, exchange ideas and expertise. So we are this year we are hosting, in addition to this webinar, a scientific workshop, a hackathon in September 28 and October 3 and 5 to 5. The event was advertised in the last few months, and it's going to be, I'm hoping it's going to be renewed in the coming years. And the second event is a technical workshop that we are hosting on site also in the late September, where people can come and learn about how we standardize data acquisition, how we standardize experiment, and also create community and exchange between the people that are coming. So that community aspect is really important. Now, to speak to the proposal cycle, so we are in the process of the second year. So last year we selected 3 projects, and the last year capabilities was centered around neuropixels and 2-photon recording and passively viewing mice. So you can see here on the top line. Our plan is to have four a selection cycle in the next in the 4 or 5 years life of the resource. Currently with the NIH funding. This year we are keeping the last year capability, but as we plan, extending our technical capabilities. This is an important year for us. It's the first time we're introducing behavior control to the experiment, and for that we enable experiment that leverage the detection of change task. So it's a task that we have built at the Institute already. We know how to train animals, and you'll be able to modify that task during the recording days to test various questions of cortical computation. Like in the previous years, we'll allow this for both neuropixels and 2-photon recording experiments. In the coming years, in the next call, we'll extend the flexibility around behavior and works toward that goal of more diversified portfolio of behavior capabilities. So that's the highlight of this year. Before we dive into this year RFP, I want to speak a little bit about what is the Allen Brain Observatory pipeline, so that you appreciate the effort that went into this. So as I said, this is a hand-to-hand data collection pipeline that is made available to you for testing questions. It starts with transgenic mice and branches on the way for different capabilities. So, and finish with data set that are made available on the web. There's a surgical suite involved, and it involves a lot of teams at the Institute that we are thankful to have developed that expertise. Intrinsic signal imaging mapping, so this is like mapping the visual cortex to know where the brain areas are located. And then we can either have mice habituated to a passive visual stimuli or being trained on the change of detection task. And then from this point on, it can branch either through a single plane 2-photon imaging system or multi-plane, multi-area imaging. So this is a variant of the mesoscope that was developed at Genelia by Daniel Fickinger and Kara Svoboda. Or the NeuroPixel recording ability that we have. So up to 6 probes are distributed across the cortex. And we'll speak a little bit about the area we can target in later slides. And then the brain is perfused. We extract that. Depending on whether we do 2-photon or NeuroPixels, we use different technique to reconstruct the brain to locate the area that we image. And then we build our product. So that entire pipeline is already working, and we are sort of leveraging this for all of this year of OpenScope experiments. This process is entirely standardized. Here we're sort of highlighting a number of CAD that were built as we sort of develop the instruments. You see that between the NeuroPixel rig and the 2-photon rig, the behavior suite underneath is exactly the same. So that allows to sort of compare experiment between recording capabilities. And we have a sophisticated coordinate system that goes from the surgical suite to the physiological experiment to precisely locate areas that are recorded from. You can see more insight, more information on this in these publications, or you can also access the hardware that was designed. This design has been made open to the community as well. There's a lot more to speak there, but in the interest of time, I'll limit to the slides. This is just a slide to highlight the multi-area calcium imaging capability of this pipeline. This also to be developed involve a lot of work, including Natalia Olovia and Dimitri Tsibulski. So this is quite a flexible platform that can position areas of interest all across the imaging implant. Here, it's a little stacked image to highlight that we can record from 4 layers, 2 set of connected cortical column, but that also can be reconfigured to have 4 areas around. The details of this possibilities are highlighted on the RFP PDF document that you have access on the web. The NeuroPixel recording was implemented by, again, a really diverse team led by Sean Olsen, involving Severin and Josh Siegel. And that system allows to position 6 probes at very standardized location in the visual system. But we have worked in the last 2 years to extend this recording capabilities beyond vision. I'll speak to that in the next slides. But you see that it allows really reproducible identification of neuronal units in the context of the experiment that you will be proposing. And if you want more information on how this is being built and the details of it, I invite you to find this publication that was published, I think, last year in Nature. Now, this is the new capabilities of this year, the detection of change behavior. So this is a behavior where the animal is trained to recognize a change of an image. So an image is flashed in front of the animal really repetitively, And then the mouse is asked to lick a lick port when the image is different from the previous instance. And so this is highlighted here, where you see that on image change here, the mouse will start to lick and continue to lick until it has consumed the reward. Again, we have a multiple publication and some are pending that describe the details of this task. And for this year, RFP will allow modification of this task during recording days. I'll speak to that in some later slides. That platform led to a number of data releases that I made available to the community. So we describe this data set as visual coding to Photon, visual coding NeuroPixel, and visual coding to P. Each one of these is associated with multiple data releases that were completed. And so this data set form a wonderful opportunity to sort of test questions that are working within the context of these experiments. And this has been certainly the case in the last, let's say, 5 years. The number of publications coming from that are using this data set have been growing steadily every year. And so this forms sort of a wonderful platform to sort of synergize the field around this data set. And now, once you have done this kind of exploration, the question is, how can we open this up to allow more targeted questions and to collect data sets that are more tailored to specific hypotheses? So this is the context of this request for proposal for 2022. To give you some context, so last year we made this exact same call. We had the webinar in September. This year we pushed it earlier so to give you more time to put together the proposals. We went through this process of selecting proposals. We'll speak to that. Carly will speak to that shortly. And selected 3 projects, 2 NeuroPixel project and 1 2-photon project. They sort of touch, when we receive more, but the one selected touch a brief sort of a range of questions. For instance, this one speak to prediction errors and comparing local and global prediction errors. This is from the team at Vanderbilt. We started the data. The process of collecting the data in January when the project was selected by the steering committee. Now the stimulus was written and was deployed on the team. We went through the process of testing this on the platform. We have already collected some data set for the team, and they were converted into NWB files, and now they started to explore some of the first units responding to their stimuli. So this is ongoing and is on track. The second project also had, we're speaking about the neurocorrelate of illusion and use this as a proxy to look at the process of binding, so for connecting visual object together. Again, in collaboration with the team, the stimulus was deployed, was written, tested. We're almost actually finished with data collection. So shortly, the team will be going into full analysis mode on this project. The last project is about looking at coupling between soma and dendrites using 2-photon calcium imaging in the context of predictive learning. And again, we've deployed the stimuli. We are about to start data production this summer. So overall, we're on track with this project, and that will give time next year to take over the next call for our projects. And so, as I said before, this will involve the detection of change behavior. It's the same slides as previously. The technical capabilities are very similar. So either 2-photon or NeuroPixel experiments can be proposed. But now, instead of only a passive training, you can leverage the behavior capabilities of the pipeline to test questions that involve rewards. So the mouse is actively engaged during the experiment, and we have a measure of it. So that's the important change. For the 2-photon experiment, that's the major change. So you see here that we've added in blue this detection of change capabilities. For the NeuroPixel pipeline, we have also worked to extend with the NeuroPixel team at the Institute the range of areas. So it's an entirely new surgical preparation that took a lot of iteration and effort. But instead of having only the 5mm window, it's essentially a full dorsal skull, semi-dorsal skull implant. And we can make holes across that implant, allowing to record across the dorsal cortex, as well as some subcortical areas. Boosts are listed here. And so we think that greatly extend the breadth of questions that can be asked with this platform. In the PDF document that you can download, these areas are listed in a little more details than here. And so I invite you to look there. If you have questions on a particular brain area, also encourage you to reach out to us, and we'll be happy to look into this. Some areas are obviously easier to target than others, and so we want to evaluate it with you if you want to do something that is beyond this. But the striatum, the hippocampus, the motor cortex is now open, and so we are quite excited by this extension. And in future years, we'll do the same for 2-photon, but this is ongoing. Now, this is the big change, it's the detection of change improvement. And the higher order bit on this is that the mice are trained on the detection of change task on the cluster, and we want to keep that constant for now, because whenever you change a behavior task, there's many ways the training can fail. And so by keeping that, we can guarantee the training to perform really well. And then what you can do is modify the task in many different ways on the recording rig that allows to introduce new components, error signals, deviations, that sort of to look at how the cortex is adapting to this change. So we think it's opened up a lot of potential questions, but this is the first time the community is doing like a behavior observatory, and so we want to do it right. So we do it step by step. There in the next 2 slides, we're highlighting some of the variants that can be done. You can change the setup image that I presented to the mouse. You can also shorten in the behavior training. We go from transitioning mice from gradings to a natural image, and you can shorten that training to stick to gradings if that's better for your questions. Also, there's a component of temporal kinetics in the task. You can modify these. There's also a component of unexpected stimuli. So as part of detection of change, we introduce at a certain fixed percentage a number of omissions. So the mice is seeing the image, and then suddenly it's a blank frame that is presented. That allows to sort of tackle questions about unexpected stimuli. And you can modify that behavior. There's also like pre and post behavior blocks that could be, you could introduce more things that the mouse will be sort of preconditioned on. And so essentially change the rules. In the PDF document, there's a long appendix that describes this in more details. And so I invite you to go over this. And if you have questions, you can schedule one-on-one with us to discuss, or reach out on the email address. Okay, so I'm going to hand it over to Carly, that's going to describe the selection process.
Thank you, Jerome. So yes, we definitely encourage everyone to reach out as much as possible for questions on this with the RFP and what is possible with our pipeline for your experiments. And this comes into the selection process. So we have a pretty unique model here where we're allowing people from the community to propose their experiments and opening it up. but still have the constraints of running it on the Allen Institute pipeline and someone else's lab. So part of our review process is trying to balance selecting applications with the highest scientific impact, but also making sure they are feasible on our pipeline and resources at the Allen Institute. So one, we've tried to really lay that out in the RFP so you guys know what kind of experiments you can propose. And then we're also checking that throughout the review process. So definitely reach out for questions. We're going to try and help as much as possible to get good applications in that are feasible. The application process is similar to last year, but there's a few tweaks because of the new capabilities this year. So again, we're going to have everyone apply with a 2-page letter of intent. The due dates changed slightly, so September 12th this year. And this is open to international applicants. This is open to traditional academic university set organizations, to individuals. Someone in the questions asked about citizen scientists, and this is again open to a broad community. So citizen scientists can be applying for this as well. And if you have questions on who can apply, what career level, we definitely encourage you to reach out. So this is a blinded process, international, any career level, any organization can apply. Just be aware we're going to have folks signing a collaborative research agreement with us. So make sure you and whatever organization you work under are okay with signing documents if selected. So just like last year, we'll submit a letter of intent. What we do first is an internal blinded review. One, we're checking that you followed the instructions in the RFP, that your application was appropriately blinded. And we also have an internal review, just making sure, is this feasible on our resources? If you included details not specified in the RFP, we might want to check and make sure we can feasibly do this project. And then the purpose of this is to let you know if the experiment you're planning to do is just not feasible at this time. The feasible letters of intent will go to a blinded external scientific merit review for people outside of the Allen Institute. And they're going to score this on kind of scientific impact. The feasible experiments that have the most impact, we are going to then invite 18 of those for a full proposal. And I'll talk a little bit more about how we're rating these in a second, but we've increased this a little bit from last year to 18 full proposals. Those who are invited for full proposals will then have about 6 weeks to write a more fleshed out 6-page full proposal that will be due on November 7th. When we receive those, just like last year, we have this, again, both an internal review with blinded Allen Institute staff who's checking with these additional details, is this still feasible? And also ranking kind of the time resource risk of this to kind of look at, is this a very complicated experiment that might take more time? Is it fairly simple? And just adding a score so we consider that. And then at the same time, external reviewers who again are blinded are going to be scoring this on the scientific impacts, how well your experimental methods answer the proposed scientific hypothesis, et cetera. And then both these are weighted to decide which collection of full proposals will be sent forward to our OpenScope steering committee, who will then look at the entire kind of balance and collection of the highest ranked full proposals to then decide on 3 final projects to be awarded and selected for this year. So they're making sure there's a good balance of the 3 selected in the end. Something new this year, because we are adding some more complicated capabilities, we want to make sure those who are proposing passive experiments or those who are proposing behavioral experiments are all have an opportunity to be selected, and we have a balance of 3 projects at the end. So something we're doing this year is splitting it into 3 different tracks. So just like last year, you can still submit to do a passive viewing experiment on either neuropixels or 2-photon, and that is track 3. That is similar to last year. They're a bit less complicated experiments for us. And then we have separate tracks for those proposing to do this new capability with the behavioral experiments. So we'll have a track for behavioral experiments on NeuroPixels platform. And then track 2 will be behavioral experiments on the 2-photon platform. And then again, track 3 is just like last year, the passive experiments there. So during both, when we're choosing which LOIs to select for full proposals, we're going to make sure there's a balance across these 3 tracks. So if you're proposing passive experiments, you're still going to have a chance to be selected for full proposals, and same with behavioral. So we're trying to get a balance across these. And also just a note, so don't just submit to do behavioral, because it might make sure it's relevant to be adding these experiments for your proposal, and it adds to the scientific impact. So same when we do the full proposal review, we're going to make sure that a balance across all 3 tracks are going up to the steering committee. So we make sure the final 3 projects are balanced across these 3 different tracks. We can only do so many behavioral experiments, so we can't have all 3 behavioral neuropixels or all 3 behavioral 2 photon, but we can always have as many passive viewing experiments as we want. So we're trying to get a balance, but we can't select too many of the more complicated behavioral experiments. So that's something new for this year, and we welcome any questions on that. One other thing that's new for this year, and Jerome, if you want to go to the next slide, we've added a small new section to the full proposal you guys are going to submit. And so within this 6 pages, we're asking for a small write-up about a diversity statement. So one of our goals for OpenScope is making sure we are increasing access to the community and broadening access across the neuroscience community. We also have a blinded process, so we're trying to work in a section of the full proposal that gives the applicant the opportunity to tell us how if their application is successful, this will be broadening access to within the neuroscience community. So we are giving room for you to suggest how your proposal, and if you're selected, will broaden this community engagement. But one thing could be noting that theoretical scientists will now have access to experimental methods they wouldn't have access to without OpenScope, or how different groups might have minimal access to neuroscience infrastructure, and OpenScope allows them to do something they couldn't normally do. So this is a new section. This is going to be considered in the full proposal stage when we're scoring this and deciding who goes to steering committee, and is about a half page or 150 words. One thing to note here is we're asking you to try and keep this blinded while writing this. And if you guys have questions on that, please reach out and let us know. Okay, one other thing to add quickly is to submit these, we're going to be using, again, the same platform of the Submittable platform. And you can find, we will be able to find the apply link through the OpenScope website. It's not currently active, but it should be soon. So don't be confused if we don't see where to apply and submit your application, but they're on the same OpenScope website. You found the RFP, there'll be an apply button, and then we'll go through the Submittable platform to submit those and do all our reviews. All right, next slide, Jerome. So what each awarded team will receive is access to the Allen Brain Observatory pipeline and expertise. For 2 photon, this means NWB files of the data of up to 90 curated, and by that we mean passing quality control sessions across a maximum of 10 mice. So we will most likely be performing experiments in more than 10 mice, but we're going to give you 10 that pass quality control. Same with neuropixels. So if you have a neuropixels project that you're proposing, you would receive NWB files of up to 10 curated, passing quality control, single sessions across a maximum of 10 mice. Applicants who are selected will also get 1 week of reimbursed visit to the Allen Institute to visit the observatory for one of your team members. So that's a part of the funding we have, is to allow you to come out and visit and see the pipeline. And the awarded teams are also going to get exclusive access to the NWB files on their project that will be posted on Dandy for 1 year after data collection ends. We have this optional embargo that we're going to give your team private access to this for the first year. If you want, you don't have to take that. And then after the year, that data is going to be open to the rest of the community. But it does give you advanced access to the data created through your selected proposal. Future calls, we will provide essential compute resource on a Jupyter Hub server. And then generally, this is giving you, helping you join the growing OpenScope community. So being part of this community that we're building with OpenScope. To the next slide. So what we expect from the applicants is just really amazing proposals proposing impactful science. So we want to see what the community can offer in proposing really great impactful science that we may not be thinking of. And we also, when doing that though, is just a reminder to propose impactful science, but feasible with following the instructions in the RFP and doing the simplest experimental design possible. So don't add more than is necessary to answer your hypothesis, but the simpler experiment, the better, so we can run it on our resources. And again, we're asking just for alignment to the RFP and those experimental capabilities at the Allen Brain Observatory. Again, we welcome questions on that if it's not clear in the RFP, but please reach out for any help if you're not sure what's possible on our pipeline. If you are selected, we're asking for commitment to the project and this commitment of time while we're running this experiment for you. So the awarded teams are expected to supply a functional visual stimulus code to the Allen Institute. So the experiment will be waiting on that. We also expect interaction between the team and the Allen Institute throughout your project, especially during this initial period where we're kind of working out your visual stimulus code and kind of initial pilots before we're up and running on the experiment. So we'll need teams that are interactive and answering any questions we have on our end. When we then are finished collecting the data, we expect the awarded teams to do the execution of their data analysis plan. So that is something on the awarded teams to have the resources to complete the data analysis of the data we hand off to you. And then finally, we expect the teams to follow up with a written report outlining the results of their analysis and efforts towards publishing in some peer-reviewed scientific journals. We're looking for teams that will continue to engage with the OpenScope community members and the agreement to disseminate these results in a public release of the data analysis. So again, this should help the broader community in the end. Jerome, do you want to do the next slide? All right, Jerome, I'm going to hand it over to you for the future of OpenScope.
So this is a future facing, a couple of slides, and we are, so what you can expect from the future calls. As I said, this is an important year. We released the behavior for the first time, but in the next two calls, we are planning to sort of extend the behavior capabilities, and this will be defined in partnership with the steering committee. And so for the two next call, we'll do that. In addition, we are also planning to extend the recording areas for 2-photon. So this is the plan that, this is the plan that we released to you last year on this exact same webinar. So we are in year 2. We are in the process of working towards behavior extension. The cloud analysis is something we'll keep working on, so as to make it available to the community. The wider NeuroPixel recording is something that we're releasing this year, so you're getting access to this. The development of the 2-photon wider access is ongoing, and we are on track. Our goal is to release it next year. So essentially, that's a half hemisphere dorsal access. It's a variant of the crystoscope preparation that was published, I think, 5 years ago. But will give access to imaging access to most of the dorsal area through 2-photon imaging. So we are currently validating this preparation. And the last activity is about the web portal. We're going to keep growing that resource. We've built YouTube videos describing projects and so on and so forth. But sort of the help is to continue to develop the community this way. So to summarize, I hope we've convinced you that this is a new and exciting model for knowledge generation in system neuroscience. We discussed in details this year RFP, and now we're hoping to answer questions. But if you did not get the opportunity, or if you're like watching this video after recording was done, I invite you to reach out, talk to us. We are sort of really available. We want to make this a success and help you draft the best proposals. So really, our doors are open. With that being said, thank you for your attention.
Yeah, thank you, Jerome. And yeah, just a reminder, we're going to be reaching out if you registered and requested a one-on-one consultation. We'll be sending you an email if we haven't already with ways to schedule that so you can talk to us for a 10-minute Zoom. Please don't hesitate to email us with questions as well. And we might be doing a second Q&A closer to the LOI deadline in September. So just be on the lookout for that. But please, again, just let us know if you guys have questions along the way and making sure your experiment can align with our capabilities. Okay.
Carly, there's a couple slides with a really important, but I forgot. This project is the results of a lot of people work, and so I'm extremely thankful for them. The steering committee, you have the listed member here. They're like influential people in the field. They help us serve you. So I want to thank them, and they've been really involved from the beginning. This is also funded by the Brain Initiative, so we are thankful for their support and continuing support. And lastly, but really importantly, this pipeline is the result of a lot of teams, from scientists, technicians, research associate managers, engineers, and animal care team. So every single box here is a team that is doing this for you. And if your project is selected, you'll see that this is really a team effort, and you collaborate with all of the components here. And lastly, the Institute is really important, supporting that platform as well. And so I'm thankful for that in particular.
Thanks, Jerome. Yeah, and this is not possible without all this help and also the NIH support of this award. And we do have, so now we have time for questions for the OpenScope Q&A for the program. And then we also have time for general questions afterwards, either for us or the Bonsai program as well. A couple questions have already come in, and I think one we touched on briefly in the talk, but we should probably reiterate, is people are asking about just clarity on the brain regions we can record from, whether it's cortex only. So maybe, Jerome, you can cover that again about the difference between the NeuroPixels projects and the 2-photon, and what is possible this year, and what might be possible in future years.
Yeah, so, well, the experimental apparatus is centered around the screen. And so currently, most of the input is visual. The task has component of like a motor components and many other sort of related components, but it's centered around vision. We're working to extending to add auditory cues in the future, but at the moment, it's centered around vision. But that doesn't mean that we can't record from areas beyond the visual system. So for 2-photon, every area within the 5mm window is accessible. So that means there's a lot of secondary multi-sensory area involved, as well as associative area in the window, including RSP or things like that. That's for 2-photon. For a NeuroPixel, because the probe is so long, you will have access to subcortical neurons, as well as because we are now enabling a more extended set of target, you can target areas across most of the dorsal cortex. So that involve somatosensory, motor cortex, a lot of potential insight about the non-visual component of the task. And you also will have access to the behavior videos, but the mouse is so you know the response in more detail. So I think there's a breadth of questions that can be tackled that are non-visual this way. I hope that that's a good answer.
Yeah, thank you, Jerome. And again, there's details laid out in the RFP on what areas are new this year. We have another questions on, do you have any experience or research on paralysis or motor cortex?
Not currently. I think we'll have to. It depends how you would look into this. If you use pharmacology, we could look into that. I encourage you to reach out. It depends on the details. Yes, again, because that has a big impact on how it's implemented. But the task has multiple components. So I guess we'll have to discuss with you, and I encourage you to reach out.
A lot of these questions is helpful for us to have kind of a back and forth interaction to see what the specifics are, and the feasibility will just depend on the experimental design you're considering. Someone has two questions. Is it possible to include treatments or changes in the diets of the animals in the proposals?
I think we should try to consider injections, but it depends on the substance a little bit, because some are control substance that are really hard for us to get and also have like a regulation aspect. But yeah, it depends on details again. The diet, I feel like it should be possible. We've looked into this a little bit last year. If you have a rationale and a strong experimental design, then what we will do is we'll reach out to internal teams and discuss the ramification of this change and provide a recommendation.
And we had someone ask another question, and we briefly touched on this in the talk, but we only mentioned a few communities that we're trying to expand access to in the intro of the talk. And then what about including citizen scientists as one participant in the community? So any thoughts on that, Jerome?
Every scientist is a scientist for me. I don't think there's a particular community that will be excluded. Again, science is the main selection criteria, and your commitment to the project. So if you have a good project and you're committed to it, try it.
Yeah, and again, this is eligible pretty broadly beyond typical other opportunities. So internationally, any career level. We had a lot of questions from grad students, postdocs before. Do they need to have their own lab? And you don't need to have your own lab established. So you can be early career, you could be citizen scientists and apply. And again, this is a blinded process where we're looking at mainly the impact and the feasibility at first, and then looking at how to increase community access. So we definitely encourage those applications. And if you have further questions, definitely reach out to us. Another question, can the behavior only include reward or also an aversive solution?
So it depends how you build aversive. So there is a board component in the detection of change that are designed to make the response disencourage. I don't think we can add new hardware, because that will mean introducing like a HEPA, if what you're thinking, because we will have to introduce this across the entire pipeline. That's going to take time to build at the moment.
And we have someone just clarifying who's doing what when they're proposing these experiments. So clarifying that the experiments are performed at the Allen Institute by the staff of the Allen Institute, and that is correct. The data collection is going to be performed by our staff and personnel here. And then what is the role of the person who proposed the experiments? So Jerome, you want to outline that, who's doing what in the roles here?
Yeah, so we collected that, you proposed the idea, you're like the guiding our experiments with the experimental design. You help us design the stimulus code or the behavior code that is involved in the experiment. We'll have to check that it's doing as you want. Then we deploy it together on the pipeline, make sure it works. We provide feedback to you on the performance. We evaluate this together, and then we run the experiment. We do the animal husbandry, animal care, and implantation, recording, everything. We package the data and we send it to you. And then from then on, we pass the stick and you have to do the analysis to test your questions. So that's the division of labor.
And we have another question coming in, just clarifying the number of mice. So the maximum of 10 mice, is that including controls? What exactly does that 10 number mean?
That's the number of mice we've collected data on. We could do more experiments to check that the data is good quality. We already have quality control involved. Is that what you meant with control? But yeah, you'll get 10 mice of data that are worthy of analysis.
Right, so that's 10 mice in the end of quality control data. We might run more than that, but we assume some might not pass the quality control, but we're aiming to get you 10 in the end of quality control data. So if your experiment includes more than that, it's just looking at how you can design an experiment that fits in with that max number of mice and quality control data you'll get out in the end. Okay. I think questions on how to be competitive when you're proposing, say, if we're adding behavior this year, are the behavioral ones going to have more scientific impact? Are you still going to be competitive if you're just proposing something similar to last year?
Yes, that's the intent of having these 3 tracks. So all of the experiment will be judged independently on their capabilities. And so if you're proposing a passive experiment, you're encouraged to do so. You'll be perfectly competitive with all the other capabilities.
And along with that, if you submitted last year and were not selected, can you submit again this year?
Yeah, please do. We've provided feedback on the selection process. Reach out, iterate, make it better.
We had a lot of competitive applications last year that unfortunately we couldn't select them all. So we definitely encourage people to resubmit again this year if they weren't selected last year. And again, with the tracks, hoping we'll still be competitive, even if you're not including the more complicated experiments. How about teams submitting that are across different institutes? Is that allowable?
Yes, multidisciplinary team is encouraged. In fact, I think it usually makes for better proposal because you introduce more viewpoint and more insight into your experiment, and that shows in the experiment in the end. So I really encourage you to gather people and try to build a proposal together.
And again, the one thing we ask is it's probably good to give your institutions, kind of legal team, et cetera, a heads up that you're applying. So if you are selected, we will be sending out a collaborative research agreement for both us and the teams to sign. So it's just nice for that not to be a surprise for your legal team on your end, and that you'll be involving both institutions if you're a team submitting and chosen in the end. And I think we're about to open up questions both for the Bonsai as well, if people want to ask on that, but we're continuing to welcome questions on OpenScope in addition. Jerome, anything else that wasn't asked we want to make sure to cover?
No, I think that was good. We had a good set of questions. Please reach out with the one-on-one. I think there might be questions that are specific to your project, and I encourage you to reach out. If you do, we'll answer your questions in a private manner, and you'll be safe. We'll not involve in the selection process, so we can be unblinded.
Great. Yeah, so definitely reach out if you guys have additional questions, either by the OpenScope@alleninstitute.org email address, or through the one-on-one sessions if you haven't already scheduled one. And this will also be recorded and on YouTube as well if people want to go back and reference this in the future. We're going to see about doing a second Q&A sometime closer to the letter of intent deadline, and looking at what format will be best suited for that. And definitely want to make sure to encourage people to be able to submit the best proposal they can if this is not typically their area of expertise. So definitely don't be shy in reaching out. I'm just seeing a few other questions that are buried in the chat, and a reminder to put it in the Q&A box if you guys have a specific question. But just someone just asking about cerebellum included in subcortical areas. And I don't remember off the top of my head if that was included, Jerome.
I don't think the cerebellum is included. So the reason is in the neural pixel, the new implant is a plastic piece that is 3D printed. And it goes from the back of the visual cortex to the front of the cortex. So the cerebellum is a little behind. And so I don't think we can introduce a probe through this implant at the moment. Subcortical structure, yeah, there are some that are currently available, not all of them. So some subcortical nuclei, tiny, and even with dedicated effort, are hard to target. And so in the PDF document, we list some of these that we think we can reach reliably. But if there are specific nuclei that you're interested in, I encourage you to reach out, and we'll evaluate it together with the NeuroPixel team.
And one last question similar on the striatum.
Yes, it's listed.
Perfect. Okay, great. Well, again, if there's any more questions, please reach out to us by email. And we thank everyone for their attention today, and are excited to kind of be growing this community. And please reach out if there's anything else you guys need help with. All right. Thank you all. And